Okay. Hello, I'm Emily Garza, and I will be presenting for me and Angelica Ramirez. And I will be presenting chapter 3 and 4, antigen capture and presentation and antigen recognition. So what is an antigen? So an antibody generator, or the substances that are responsible for inducing the production of proteins and reactive lymphocytes that have the ability to fight against foreign macromolecules when they enter the body. So these epitopes, they are actually part of the antigen that are recognized by antibodies, and they're present on the surface of these antigens. They are small in size and they may differ in conformation. So in this picture, you can see the epitope is right here. It is on the antigen, and this is the part where the antibody recognizes the antigen. So how do B and T lymphocytes differ in the types of antigens they recognize? Well, B-cell receptors recognize proteins, polysaccharides, lipids, and nucleic acids in soluble form or cell surface associated form. B lymphocytes can recognize antigens in their native conformation. T-cell receptors, however, recognize peptides presented by MHC molecules or major histocompatibility complex molecules. So types of MHC. So there's class 1 and class 2. Class 1 can be found in all nucleated cells and present to CD8 cytotoxic cells. Class 2, meanwhile, are present on antigen-presenting cells, or APCs, like B cells, macrophages, and dendritic cells, and they only present to CD4 T helper cells. The MHC molecule's function is to primarily present the antigen epitope to these T cells. And here in this picture, you can see MHC class 1 right here, which presents to CD8, and class 2, which presents to CD4, which we said in the previous slide. So the antigen processing pathway. So class 2 converts protein antigens into vesicles of APCs into peptides that bind to class 2 MHC molecules for recognition by CD4 T cells. Class 1 converts protein into cytosol into peptides that bind to class 1 MHC molecules for recognition by CD8 T cells. And in this photo, you can see how they convert the protein into the peptides, which will then bind to the MHC, which can be presented to the CD8, and this would be the class 1 pathway. In class 2 pathway, they are put into these vesicles, converted into the peptides, which then bind to the MHC and then present to CD4. So the role of dendritic cells, which are the, and I said in the previous slides, which can be these APCs. The dendritic cells, the objective is to capture protein antigens of microbes that enter the cell and transport to peripheral lymphoid organs, lymph nodes, and the spleen for T lymphocyte recognition. The immature dendritic cells will reside in tissues like skin and form a network of cells. And the mature dendritic cells will be found in T cell rich areas of lymph nodes and the spleen. Here you can see the immature and mature into, when undergo maturation, can undergo to mature dendritic cells. So cross-presentation. So parts of cells infected with intracellular microbes are ingested by dendritic cells, and the antigens of the infectious microbes are broken down and presented in association with class 1 MHC molecules to APCs. Then these T cells recognize the microbial antigens expressed on the APCs, and these T cells are then activated. CD8 T cells recognize class 1 MHC associated antigens, and the same cross-presenting APC may display class 2 MHC associated antigens from the microbe for recognition by the CD4 T cells. So the development of lymphocytes. So B or T lymphocytes will begin their development in the bone marrow, where they will start the maturation process. Once they become thymocytes, during the maturation process, they can also be found in the thymus, hence the name thymocytes. After completing this process and becoming mature cells, they will then travel to the peripheral lymphoid organs or tissues. So the selection that undergoes during different stages of maturation. So the process of lymphocyte maturation starts with proliferation, where pro-B or T cells will express a part of the pre-antigen receptor. If this fails, it'll undergo negative selection and apoptosis. But if it succeeds, the cell undergoes positive selection. Next, these B or T cells will complete the receptor, therefore expressing the whole receptor chain. If this fails, it is negatively selected and once again undergoes apoptosis. If it succeeds, however, positive selection occurs. After this stage of selection, the now called immature B or T cell will express the complete antigen receptors. So you see they created part of it, they will express part of it, and then if they're positively selected, they will express the complete receptor. And then the immature B or T cell will undergo another selection, this time to test the reaction to the MHC on APCs. If the cell either one has no recognition of the MHC or has a strong antigen recognition, it will be negatively selected and undergo apoptosis once again. But if the immature cell has a weak antigen recognition, it will go through positive selection to become immature B or T cell. So in this picture, you can see how if it does undergo weak recognition, it will undergo positive selection. And this one is for the class 2. This one's class 1. And if it does not recognize it, it actually doesn't undergo negative selection. It undergoes apoptosis or death by neglect, or basically just failure of positive selection. But if it undergoes too strong of a recognition, in this picture it binds twice, then it'll undergo negative selection. Negative selection is important actually to ensure that not only do mature cells express functioning receptors that can carry out antigen recognition, but also that they are self-MHC restricted. It will contribute to the maintenance of self-tolerance, basically. And here's just a full photo of the process where it first expresses partially of the receptor, then the full receptor with its with the CD4 or the CD8, and then it'll undergo the weak recognition, no recognition, or too strong recognition. So how and why is the vast diversity of receptor structures in the lymphocyte repertoire generated? So the formation of the genes that encode for B or T antigen receptors are initiated by somatic recombination of gene segments that code for the variable regions of the receptors. Because there are not enough genes in the body for every receptor to be encoded by a different gene, B or T lymphocytes go through different cycles of proliferation and expression of antigen receptor proteins by gene recombination. And that is it. Thank you.